unfortunate. Simply unfortunate they have mold in their name, but we hope that everybody will remember that they're not slime molds. Tyson and Pam and Ryan, and to a much lesser extent me, are very involved with trying to document British Columbia's slime molds. And to complete my introduction to my friend Tyson, I thought a bit of poetry might be appropriate, certainly in terms of limericks. Gather round, all you young fellers, to celebrate our guest, Tyson Ehlers. He has tales to be told of these so cool slime molds and of wet reproductive flagellar. 3 cheers for the myxomycetes, whose weirdness inspires heebie-jeebies, and yet whose intelligence and inherent elegance inspires our ardent eccentricities. Please welcome our guest tonight, Tyson Ehlers.
Bravo. I mean, I don't even need to give a talk now. That introduction was brilliant. First of all, yes, they're not fungi. That has to be said. And I never found a polite way of saying I was going to get angry that I got invited to speak with mushroom people about something that's an entirely kingdom. But you so eloquently said that who else would speak for them if not us? And of course, you bring in the poetry. I'm so glad that was recorded. That was it's the thing about slime molds is they're they bridge all the aesthetics. They're just these exquisite life forms. They have no practical uses to us. Sure, they figured out mazes that emulate the Tokyo subway system, and they've taught us a lot. But unlike non-timber forest products, and my background in foraging and how we all get into getting to the woods in our various ways, for me a big attraction was, can you eat it? Can I make money from it? And I did and still do those things with things I forage. But the only thing I forage with slime molds is time and appreciation of nature. And I think that's wonderful about them. They don't have any economic value, real economic value. So I guess I should open up my sharing screen here and see if I can navigate through the world of Zoom. What I've got lined up, I'm little disclosure here. I came down with a nasty flu Friday. Spent the weekend in bed when I should have been prepping the slime talk. So I got a bit of a cough that I might have to take breaks and deal with. This cover slide is a slime mold taken with a slightly post-processed picture of under UV light, which I got a bit obsessed with doing. Slime molds aren't all that UV responsive, just in certain phases when they're quite watery, and certain species. So I do get a little artsy with my photos in that regard. Thank you, Andy. Thank you, Melanie, the president, the new president, and thanks to David Waldy, it's German sounding, past president, who persisted in getting me to give this talk. And as much as I felt not really that qualified, it's just, I guess I'm more qualified than most because I've been obsessed with slimes. My background is in mushrooms, as Andy mentioned, and basically everything. I really study those things that get overlooked. A lot of bugs. I do a lot of bug stuff, and but the slime molds have really taken over as a main focus in my research, and I see a need for it. We don't have any. We didn't until we started last year. Myself, Andy McKinnon, I guess 2 years ago, Pam Jansen, and Ryan Durand, and there were others, Kent Brothers, who started was in the original team, and others on the periphery have been involved in let's do something about this. Let's get an inventory going. And so Andy, with his connections to the Royal BC Museum, has coordinated a book deal. So we're going to write a book, and this will be the title: Slime Molds of British Columbia. And so I just need to acknowledge that this is a small group of us doing this, but we're passionate, and it's real. It's happening. So what is a slime mold? Well, first of all, not fungi, distantly related. If you go into the whole eukaryotic tree of life, these things are in a completely different kingdom, the Protista. And I guess with the new, depending on which classification scheme you follow these days, it's the Amoebozoa. They're amoeboid eukaryotes, which produce macroscopic fruiting bodies with a relatively complex structure. And within the Amoebozoa, the phylum is now established. And this is only 2 years ago. I had a different name for this 2 years ago, but now we're calling it Eumycetozoa within the supergroup Amoebozoa, which, if you follow these phylogenetic trees, you could see how distantly related they are, but at least in somewhat of a common ancestor to fungi and animals. And they're unicellular, as amoebae are, but they form these macroscopic fruiting bodies and structures that also have interesting behavioral aspects. They've been demonstrated to solve mazes. They learn. There's all sorts of YouTube videos and other channels on that. That's not the focus of my talk here, but it is the headline catching aspect of slime molds. So there's a bit of a background on them. They are like fungi were about 30 years ago in terms of our knowledge and where the world was at with understanding, how fungi have become popular. Well, fungi or slime molds or myxomycetes are becoming exceedingly popular, at least in my circles. And there's a bit of a myxo revolution happening. And I like to think that Andy, Ryan, Pam, and myself are at the forefront of this. One of the founders of myxomycetology, at least in North America, is Thomas McBride. And I'll read this out. I know it's annoying when somebody reads while you're looking at it, but I just love this quote. Their minuteness retires them from ordinary ken, but such is the extreme beauty of their microscopic structure, such the exceeding interest of their life history, that for many years enthusiastic students have found the group one of peculiar fascination, in some respects at least, the most interesting and remarkable that falls beneath our lens. Now, Thomas McBride was a pioneering North American mycologist around the late 1800s, early 1900s, president of the University of Iowa, I think. And, he was a mycologist. He was a bit of an all-around naturalist. In his book, The North American Slime Molds, which has a great old-timey title, he quotes the German, the translation I got from the German is, "To approach again wavering figures who once showed themselves early to the dull gaze." And that kind of sums up slime molds. So Thomas was into this. Oh, I got into it. I don't know. I see similar forms in nature, and I guess I was being a mushroomer, kind of bored in the winter months around here in the interior. I spent a lot of my time cross-country skiing. I like old forests, and so I got interested in these pin lichens, which are also obscure, understudied. They're in the sub-millimeter range, and you can see This is Sclerophora amabilis, which was apparently a worldwide IUCN red listed species. I have found dozens of times, but peculiarly only on the very oldest cottonwood trees, and seemingly only in the Slocan, the West Kootenay area, near rivers. And so there's a similar form here. We've got a stalk and a cap. In this photo here, there's a myxomycete with some smaller pin lichens in the background. But these pin lichens, they opened up doors to new habitats at smaller scales, peering around at the bark cracks on large old trees and old forests, places like to hang out. They tend to go unnoticed by the casual observer. So too with slime molds. I soon found out, unlike pin lichens though, they're ephemeral, and maybe that's part of their appeal. It's like mushrooms in that way. It makes them more challenging and all the more interesting. So we have to have the obligatory life cycle diagram. There will be a test at the end. How they start out from a spore that hatches essentially into these swarm cells, which may or may not be flagellated. Swarm cell is the common term. Amoeboflagellates is another term for it, and those will join together if they feel the love, and they'll form a zygote. And that zygote will then continue to divide by mitosis, but not having cell divisions. So it forms one very large cell, and that cell grows and forms a plasmodium. The plasmodium is the main vegetative stage. It cruises around and munches on bacteria and things, and in the right conditions, it will form these fruiting bodies of many different forms, which we will discuss later. And that's where the meiosis and the sexual reproduction occurs, and then they make spores. At that stage, they're very much like fungi in terms of structure and function. But they do this really cool thing. Some fungi do this as well at analogous stages. They can go dormant. So at the swarm cell stage, they can form these microcysts, or as we more commonly see, because those are microscopic, plasmodium can just dry out. And I've collected lots of desiccated slime mold plasmodium and then rejuvenated it in moist chambers, and it comes back to life. So it's part of the reason for their success. And they've been around for, I don't know what the number is, 1 billion years? Let's say 1 billion years. And the other interesting thing, they found fossilized, they don't fossilize well, but they found them preserved in amber so many hundreds of millions of years ago, and they've looked at the species, and they haven't changed. It seems like there's only about 1,200 species worldwide. It seems like they just did it right from the start, and they didn't really need to change much, or at least they're changing very slowly. So we get into the classification from the Amoebozoa. We have the phylum Eumycetozoa, and there are 3 broad classes. And I don't pronounce these words very well because I read them more than I say them, but let's just call them the Dithiostellids. These are cellular slime molds which don't have a plasmodial stage. They're too tiny to be observed in nature. I won't discuss those in this talk because they're a subject of laboratories. The radiomyxomycetes used to be lumped in with the myxomycetes, but we now recognize them to be distinct evolutionarily on their own, and they only have a single genus. And their main difference is they produce the spores externally, and have a slightly different life cycle. But for all intents and purposes, they look very much like what we would consider a myxomycete. And then we get to the myxomycetes, which are the broader class of plasmodial slime molds that produce the macroscopic fruiting bodies with the spores and such that we're used to looking at. There's currently about 1,200 species worldwide. That number seems to go up, considerably every year as people are studying them more. And in British Columbia, we've got, I think I'm jumping the gun here, but we've got about 200 species. Whereas prior to our work, there were only about 100 species that were documented. So here's what a plasmodium looks like. There's many different types and colors. Different groups have very long names for the type of plasmodium they produce. But essentially, these things are single cell. That's the crazy thing about them. They can get up to a meter in size. They can move up to 4 centimeters an hour. And within them, there is cytoplasmic streaming. So they use hydraulic pressure to expand their cell walls and then retract, and constantly searching out new food sources and then waiting for the right conditions and the energy required to produce fruiting structures. I love this quote from Lucy Jones. Several people have shared this article with me. It's Creatures That Don't Conform. Neural rivers of xanthic goo, just like the veins of our bodies and the vessels of our eyes, and the branches of trees, and the clouds above, and the dendrites of galaxies, blebs packed together, river networks of slime, fan and spread, slugs, worms, springtails, and spiders attend. It abides. Love that. So much like what we see. Okay, click, there we go. So the types of fruiting bodies. Now, just like fungi, there are different, multiple names for the same things. So we're struggling with this in our book, even what to call the fruiting body. I don't like fruiting body so much, it's just so generic. Fruit, we're still a little bit stigmatized from fungi being lumped in with plants for so long. Now we've got the same problem us myxomycetologists feel about using fungi terminology to describe myxomycetes, but that's what we're stuck with. So we've got sporophores, or I like myxocarps. It just has a nice ring to it. Carp is just fruit. It's the fruit of the myxo. There are 4 main types of fruiting sporophore structures. We have the sporocarp, which has your classic stalk with a stem with a cap. And then the plasmodiocarp are when the plasmodium just sort of freezes in place, breaks up into little chunks, and makes these amorphous, almost amoeba-like structures at a macro scale. Then we have an athalium, which is one large mass. Doesn't have it's not composed of individual sporocarps, whereas a pseudoathalium has that individual sporocarp structure still maintained. So collecting slime molds. Again, Thomas McBride, he said it so well. To study slime molds and know them, we must go out of doors and be content to stay out of doors for a day at a time sometimes. The laboratory may help us, to be sure, but the lack of it need not greatly hinder us. In moist and mossy woods by perennial springs and streams, these things are at their best. Truer words couldn't have been said. And of course, one could say the same thing about mushrooms. It seems particularly apt for the myxomycetes. And this is where we go, and this is why we study slime molds. We don't get very far. You know, we find one log like that nice old log that Ryan Durand is collecting from. We'll spend half a day on that log sometimes. When they're really on, gosh, it's worse than mushroomers on a foray leaving the parking lot. We can be quite busy for a while in one spot. And you'll notice a trend. You think it's bad with mushrooms. Well, slimes, you really got to get up close, and you don't want to disturb them too much, so you end up in all sorts of awkward positions. And you have to be brave. You have to swim through the Devil's Club. If anybody is familiar with that plant, which most of you are from the coast, there's a trick to it. And it really helps when there's an incentive, like a tasty looking log in the middle of it. And you might get rewarded. Maybe not quite the, proud fisherman shot that you're used to seeing, but that is probably something really cool in his hand. And then photo documentation is key. We essentially scan the world with our little TG6 Olympus cameras, or I use a magnifying glass. I don't know if I'm quite happy. I don't know if you can see if you're still looking at this screen, but I've got the nice LED magnifying glass that I pack around, a good sharp knife, and clippers. Some really good clippers that I carry for clipping twigs and branches they may be on without disturbing them too much. A chisel to get into some of the harder wood. You want to collect the substrate they're on. You don't need a lot, but you want to have enough of the fruiting bodies, ideally that aren't moldy and that are at the perfect stage. So there's a lot of crouching, a lot of poking around, and just about anywhere. Mostly it's on dead bark, dead wood, on forest litter, dead vegetation, living vegetation, moss. They're kind of everywhere. So here's an example of cutting a piece of wood and just make out the little slime molds there on that wood, and that's about the size you get. And then I like to wrap them in wax paper and put them in my little tackle box. That keeps them from bumping around and keeps the ones that are really sporulating a lot from those spores getting everywhere, and that can mess you up later when you're looking at them under the microscope. Quick field label, and then ideally you would the classic long-term storage method is to glue your substrate to the bottom of a little matchbox and then store those, because they are quite delicate once they're dried out. And I haven't got around to that because it's not very time efficient, and I don't have a lot of little boxes, so I just try to be careful. They dry out very well. It's more the substrate that you need to worry about. So I just dry them at room temperature on various racks and places in my house, and then I'll look at them under the microscope, and then they'll get labeled, and I just put them in plastic bags and try to be careful with not bumping them around too much. And if you find them immature, they're starting to form fruiting bodies in the plasmodium, you can collect, and depending on what stage they're at, you can keep them in a moist chamber or a slimarium. And in that environment, you can get them to grow out more. You leave the lid open a little bit. You keep them in a cool, dark place. They hate light, but they want a little bit of light to start fruiting. And here I've fed them some oats, and you can see they've moved and they found the oats. And I think slime molds are non-binary, so I think they adopt the pronoun they. And I just named them all Pat because it's an androgynous name. So here's Pat. Pat's found some oats. I didn't know what Pat was. I collected Pat from a log that was starting to freeze, and Pat came back to life. Turned out to be, took me 2 years, but I got Pat to fruit, and Pat turned out to be Adhamia utricularia. Kind of cool to finally get something to work. Then we get into these nivocolous myxomycetes, which are, I think, some of the coolest because they take you to the most cool environments. So we're supposed to be doing terrestrial ecosystem mapping here, but that's what happens when you get two myxomycetologists out there doing that. We spend a lot of time on our hands and knees looking at a different layer of the environment. And so right at the edge of the fruiting of the melting snow is where these things will fruit. And so at low elevations, you got to go early in the spring, and then at the higher elevations, you can find the nivocolous. Nivocolous just means snow loving. You can find the nivocolous species right up until September. here's a low elevation. This is in April near Valley Bottom last year. And man, we got some cool things. This is a clear cut too, so it wasn't the most spectacular environment, but lo and behold, we found new things, some new species to BC right there. And that's Rufus of Winlaw. He helps out. Again, this is last year, just along the river in April. So if you look carefully under the melting snow on a log, you might see something, get an eye for these things, and that turned out to be Trichia sordida, the nivicolous species. So now we'll discuss the anatomy of a sporocarp. Okay. Examining the sporophore. Really handy to have really good photographs at all the different stages, because these things change quickly, and until they mature, we can't really identify them, many of them, without having mature features to look at under a microscope. And so for doing measurements, I use both a stereo and a compound light microscope. Any of you familiar with doing fungal microscopy would be familiar with the same processes, but imagine you're just dealing with things that are less than a couple millimeters, so you really need that magnification. So with my stereoscope, usually I'm just at the 2x objective, and then I have software that hooks up a camera to it, or I just use the ocular measurements to just for anything over millimeters or half a millimeter, I use the stereoscope, and then for anything in the micron level, of course, I go to the light microscope. But first thing I'll do, if I have a sporocarp, I'll put it under the stereoscope, and I will get a bunch of them to get an average, look for the tinier ones and the larger ones to get an average or a range of stalk lengths. And these are the key things you want to know, that what the length of the stalk is, what the length of the sporotheca and the diameter of the sporotheca are. And those ratios, the stalk to the sporotheca, can be important determining characters. And then depending on which species you have, it may or may not have all of these features. And that's what we're going to talk about now individually. So the sporotheca, also known as a sporocyst or a sporangium, is the primary part of the fruiting body that contains the spores. They come in many different shapes, sizes. You want to note the color, the dimensions, and whether it's stalked or is it globose, cylindric, pulvinate. And so here's some examples of different sporotheca shapes. See, very long, skinny, very globose, pulvinate, cylindric, ovoid. Here's a ovoid, a little wider on top than the bottom. And the stalk is a key feature in myxomycetes evolution and in determination. So the way the stalk is built, it's turning out to be kind of the deal in how these things co-evolved. So we have Trichiales, which have these thicker stalks covered in a gelatinous sheath. Then we have Staminitis with this long fibrous stalk, Lepidoderma, well, Derma now, this coarse corrugated stalk. And some stalks are filled with these round cells, not spores, but these round cysts. And here's one under a stereo microscope showing it's shiny and tapering. And then here's one just showing the granular contents. Peridium. It's the exterior sheath, the membrane that covers a spore mass. And another key character, they all have a peridium. It may be single, or it may be up to 3 layers, and it may be limey. And if you read a lot of the keys, they'll say, is the peridium persistent, or is it evanescent or fugacious, which just means it disappears quickly. And the nature of its dehiscence, that is how it how it breaks apart. Is it random, or is it in some sort of a pattern? And then under the microscope, we're going to look at the texture and the color. Here's a reticulated iridium. You can see all these maze-like ornamentation to them. Here's a limey iridium with another iridium below, another layer. It's iridescent. And then here's another one that's opening on these preformed plates. And now we get into the more microscopic characters. This is the Capellicium, and the Capellicia, not all species have it, but it's a very common feature that is very diagnostic for slime molds. And this is where you really need to get into, up to around the 400 times power light microscope to start looking at closely, and even 1,000 times for more micro features. So it may be ornamented with these rings, have these spirals. Here's a pencilate one where the threads open up. And then we have these, branching. Is it branching or is it simple like this one? And if you look at this one, there's these tiny little spines on it, on the spirals. And we have this anastomosing or reticulated pattern of another kind. And the color, we're going to measure the thickness, the diameter of these threads. And then here's a Physarum, where you can see the Capellicium is these little clear tubules, and there's these nodes in between, large lime nodes. Columella, not all species have. There's a true columella, which is this extension of the stalk that goes up into the sporotheca. And then there's a pseudocolumella, which is a blob of lime at the base, and it's not attached directly to the Capellicium. And so we're going to measure that. We're going to measure the diameter sometimes, and more importantly is how far up into the sporotheca does it. This one only goes halfway, and this one is a really distinctive pattern to it. It's sinuous, and thus the name. This is Comatrica. This is one of the longest names out there, and I still don't pronounce it. Comatrica sinuato columella. Comatrica sinuato columellata. Say that 10 times. And then we're also looking at how does the Capellicium branch off of the columella. That's very diagnostic. And then lime comes in different forms. It can be granular, spheres, or it can be crystalline. And that's a division between two major groups. And then the coating on the exterior, is it eggshell-like, or is it is it wrinkled? And then within the Capellicium could be just composed entirely of lime, and it's the nature of the shape of that Capellicium formed by the lime that we're interested in for keying out our species. Hypothallus is found at the base of the sporocarp, and it's just where the plasmodium decided to make a fruiting body. And it can have various forms and textures, and it may be dull or brightly colored, thin or thick, and delicate or coarse. Sometimes it's really obvious, and thus the slime name. And sometimes it forms a disc at the base of the stalk. This is under light microscope, but that means it's singular to the individual sporocarp, and at other times it's continuous between sporocarps. The hypothallus, another important feature. Now, Ceratiomyxales is the second class. Remember, we have the dictyostelids, we have the Ceratiomyxales, and there's only one species in BC. There's only one genus, 3 species worldwide. The other 2 are tropical, so we don't get those here. And but what we do get is a lot of Ceratiomyxa reticulosa. And so you could see this spores, you can just barely make out in this photo, are sticking out right off the appendages of the main fruiting body, not internal. So exosporic, at any rate. I call them the bullies of the slime world. Where I live, they are, it is prolific in the spring, and it forms masses of plasmodium that just drowns everything in its path, and it can happen overnight. I've seen insects drowned and various critters just consumed by it. I think it's a study in itself to see if there's actual slime wars going on out there. If the plasmodia are where you find one slime mold, you generally find others. So I'm curious how they all get along. You know, do they all just groove along on this log together, or are they competing? And why is Ceratiomyxa sometimes yellow and white? Why does it have these different forms? I don't know. I need to look that up. But here's some examples of Ceratiomyxa plasmodium being a bully. It's just growing over that poor little Pinellus and just totally coating it. Here's a snail, you know, or a slug, and I've seen snails too. They're just resting overnight, and I photographed them like this. You know, I'll go back a day later, and they've gotten up and moved along, but they don't move very fast, and clearly the lime mold plasmodium moves quite quickly. And then they used to call this poroid form Ceratiomyxa porosa or fruticulosa var porosa, but it's all just been synonymized. It's just another weird thing about this species that makes all these different shapes and forms. Again, just playing with UV light, Ceratiomyxa is quite fun with that. We get into the class myxomycetes, which are the plasmodial slime molds that produce the macroscopic fruiting bodies with complex structure. And these, we have about 1,200 species worldwide. We're now up to about 211 species in BC. This will be the large focus of our book. We're, I mean, I've got hundreds of specimens still unexamined, and so does Ryan. I'm sure Pam has quite a few that we go through these things because we've started collecting everything, and we're finding species that are new to BC and at least in one case new to science. So our BC's a big place. Slimes are small and they're everywhere. So undoubtedly we're going to find a lot more of the 1,200 or so that are in the world that we know of. So we have these subclasses within the myxomycetes. I never pronounce these words, and nobody ever does. We just read it. But let's just call them the bright spored guys, and then we have the dark spored guys. And within those are morphological classifications that are holding up quite well with DNA. Now there's some exceptions. There's some fairly dark spored light spored group, and then we have some lighter spored guys down here, but genetically that's a fairly good solid division. Within the bright spored guys, the order Cribrariales, these are frustrating because they're really hard to identify. And many of them are very tiny. This one is easy to identify. This is the Japanese lantern slime, one of the few that has a nice apt common name. And this is Cribraria cancellata, and then Cribraria vulgaris with another, I believe, a different species fruiting amongst it down here. And you can see the changes they go through. They have lime, but it's not a limey coating. It's embedded within the microscopic structure. Here's Cribraria minutissima, which I found just by scanning a piece of substrate I collected this year that had never been documented in BC. Well, we added it to the list this year. But these things are about 0.5 millimeters tall and really hard to photograph. So I don't have good macro photos, but under the microscope, this is what they look like. And then in the structure of the peridial net, remember, these guys don't have a true capolitium. The peridium just breaks up into this coins at the birdcage slime mold, which I quite like, and they can form these massive fruitings. I have this old wains, the remnants of some timber cutting, a pile of wains that have been lying around for about 20 years, and I go back and visit these guys every year, and find some large logs coated in this cribaria. Not to mention there's several other species that I find there. Just part of my going to check mail walk, may as well check the wane pile. And then another one that I've found more that I started to look at, collect a piece of substrate for something that catches my eye. That's large, like a couple of millimeters. And then there's these tiny little things that are about 0.3 millimeters. These little wrinkled domes, and it turns out this is a slime. It's Lycia minima, and it has a really cool caprolithium that split, or a iridium that splits along these preformed plates, it opens up, and under the microscope, that peridium has the margins lined with these warts, and then of course the size of the spores. Funny, I didn't have a slide on the spores. I missed that. Anyway, spores are important. Then another order within the bright sport is the Reticulareales, and we have nine species in the Reticulareales, and these are some of the ones that are most commonly observed. They form large athalia or pseudoathalia, and the classic is the wolf's milk slime mold, Lycogala epidendrum. Then we have Reticularia lycoperdon, something I'll see, in a forest fire zone. So you're out looking for morels, and you see this fleshy mass on a burnt log. That's that guy. Inside, when it's younger, it looks quite fleshy, and then of course it turns into this mature, dried out spore mass. But this is maybe our iconic one. This is Lycogala. I used to just say, Lycogala epidendrum, because worldwide up till 2 years ago, that we only had one species. And then this bloke from the Ukraine, Dmitry Leontiev, who's one of the world experts, he started looking at them a little closer, and of course found some morphological differences in specimens from across the world. And he's got up to 40 species now under Lycogala. So we can no longer confidently put that one name on it. And if you study fungi, you're used to that. What you thought you knew changes by the day. But here's a cool shot. I like this. It's like, from space. This is a giant Lycogala, giant relatively to these little cribaria fruiting amongst it. And here's Lycogala fruiting on an old Fomentarius, it looks like. I'll just threw this in because this is really what it's like when you're trying to find slime molds in the Kootenays in the spring. You're getting photobombed by mosquitoes pretty regularly, and it's one of the challenges of trying to stay still, crouched under a log, photographing myxomycetes in June in the Kootenays. And then we get to the order Trichiales, which is a large group. We have 2 families, 10 genera, and 46 species documented in BC so far. And these are, again, ones that are commonly seen. And most people would see this as I did when I first started knowing that, any little orange light bulb slime mold has got to be Trichia decipiens, which is, I don't know, has a common name, forget that one. But it's now, as of last year, Hemitrichia decipiens, and you don't know until you see it mature what it really is, because there's a bunch of different Trichiales that are members of the Trichiaceae that can have that same look when they're immature, just like the little springtail sitting on it. Excuse me. One of my favorite, I don't know how many favorites I got. I think this is a real looker. This is Prototrichia metallica, a real disco ball of a slime mold, and it has a super cool capolitium. Threads are it's called pencillate, and it just makes that really neat opened up pattern there. the capulicium in this species attaches to both the iridium and the base of the Sporotheca, so it bridges the gap inside. It's not all free floating like in other trichias. And again, the variation within genetically related species. This one's a plasmodiocarp, Plasmodiocarpus species, Dianema corticatum. Always a thrill to find. And our third species in the Dianemataceae is Dictydiathalium flumbium, very fairly common, although I've only ever found it once, and it was just last year. So these are the only photos I have of that one. Common worldwide, and I guess that's another thing I should have mentioned about slime molds is that the same species are found across the globe. They're cosmopolitan, many of them. Some are more regional, endemic, like some of the tropical species. We know there's some in in the southern half of the world that don't show up in northern temperate areas, and vice versa. And then there's the nivicolous ones that really depend on snowy climates. So it's kind of fascinating things. You can look at something and it's genetically and morphologically identical to species from remote distances. Now we're into the family Trichiaceae. Some examples: we have Arceria denudata, Metatrichia floriformis, real stunner, that one. It often forms these rosettes, all sharing the same stalk. Trichia scabra, Hemitrichia caliculata, Trichia varia, maybe the most common one in my cottonwood habitats around here, and then Hemitrichia decipiens, as we talked about earlier. Just very colorful. The Trichiaceae are very they're crowd pleasers for sure. And I got a little brag post on this one because this thing I spent a long time. I found it in November 2021 on a log in the Slocan Valley, and then I looked at it. It just looked like an Arceria. I posted online, and some knowledgeable peeps said, yeah, but that capulicium doesn't look right. I went through every monograph of all the Arceria in the whole family, which used to be Arceriaceae, which is now in the Trichiaceae, but at any rate, couldn't find it. And I got wind of a researcher in Estonia who's doing a monograph on the Arceriaceae, and I sent her a sample, and she ran the genetics on it, looked at it, and she's like, yeah, it turns out it's a new genus. So that doesn't happen every day. You find a new genus of something, and we did. And it's so far the type collection is well, just over there in my office, but it's a single log in the Slocan Valley. So we're going to write that up. Another one that was probably way more common, but it's so tiny. This is growing on a little Cephalosia liverwort that grows on very old rotten logs in very old moist conifer forests, and it typically fruits with other species, some Lamphodermas and some larger things anyways that are more visible. And yeah, I was looking through my Lamphoderma collection, and I noticed these guys, and I've heard about this tiny little barbiella that only fruits on liverworts, or is known to, and sure enough, that was it. It was pretty easy to key out, and it was the first record in the Kootenays. And now I've since found it in two other places, Glacier Park, and then up in northwestern BC, only by scanning a whole bunch of substrate that I collected from these old logs. So yeah, first record in Canada, and now we've got several. And then we get into the order Physairales. These are the ones that have lime in them. And the myxocarps, or fruiting bodies, they vary from small sporocarps to a relatively large Athalia. Highly variable. Physairales, it's a very large group. In BC, we have 5 families, 14 genera, and 102 species. Ryan has spent a lot of time. He's Mr. Physairales. You know, if you were wondering, you hear the nickname, that's him. He seems to find a lot of these. Pam has spent more time with them. I've spent less time with these guys, but I've got a few in my collection, and so I'm only sharing the photos of the ones I have. And then we get into within the Physareales, we have the actually I shouldn't say that. The Physareales is the order. I have a lot of ones within the order, but within the genus Physarum, I do not have that much. We have 5 genera, so the Diderma, Dimium, which used to be Mycological. Crustacea is a really common one that gets mistaken for Fuligo, which is the dog vomit slime mold. This one is, I think, dog 6 slime mold. You want to use common names. And these all share the same feature in the Physareales, that they have lime somewhere in them. And in the family, I should have said it, within the family Didymiaceae, they have lime. Within the family Lamprodermataceae, they do not have lime, but they have really cool structures. And this is the group I've spent more time with. A lot of them are nivocolous species, and they're some of the showiest, prettiest, most exquisite, just drop dead gorgeous life forms on the planet, really. Here's Lamproderma columbinum. You know, as Lucy Jones so poetically put it, like opalescent spherules in crinkled sweet wrappers. Love it. And this is Devil's Club. And here's the Lamproderma ovoideum fruiting on Devil's Club spines. It's always worth looking. Actually, Devil's Club is my new favorite medium, just after the snow melts, to go poke around the Devil's Club, or get around the Devil's Club. And just to show you some of the changes that these things can go through, you can see why it's really hard to identify. You see a picture like that. It can be many things that turn into that, which is Lamproderma cereoides. And these are from my favorite log, my new favorite log. Because it's on my property, I get to go down and visit it. So every couple of days, last year I took a shot, and that's the sequence of development. There, that's my new favorite log right there. And you can see the colony of Lamproderma here. Only the Lamproderma, there's Didymium, there's Lycogala. You know, there's about 7 different species of slime molds I get here. And bonus, it's got Hericium coralloides coming out, which you know, I'll take for dinner after my photo session. For the family Physareaceae, we have 5 genera, 48 species in BC so far. And these guys all have lime. Some of the ones that are commonly seen, Leocarpus fragilis. This one has a common name, insect egg slime mold. And you can see inside, I was reading the wrong thing. You can see inside there's a limey capillicium with little nodes, and same with the Physarum. You can see the little lime nodes inside, and the Craterium. So they all share that character of these limey capillicium. And here's a really charismatic, common worldwide species, Adhamia utricularia. Starts off as these bright yellow little spherules hanging from delicate strands. And this is the one I grew in my Slimarium this winter. I collected the Plasmodium that was growing on an old polypore, and that pretty much narrows it down to a few species that are known to eat fungi. You can even see an old polypore here, and this Badhamia is starting to fruit. It's quite a canary yellow, then it goes through this really cool blue-gray stage, and then it develops into this iridescent white Iridium, and very distinct growth pattern. It can be sessile, and it can look quite different. So it's not a gimme to identify, but if it looks like that, it's you know what you got. And another pet group of mine, because I'm really into these nivocolous ones, is Meroderma, which I only really learned recently in the last few years that I successfully keyed it out. When you look at the capillicium of Meroderma, outwardly it looks very much like Lamproderma. There's a few key characters, and number one, the columella is quite simple, and the capillicium tends to branch off the entire length of it, and it never goes more than halfway up the full height of the sporotheca. And the threads of the capillicium are funnel shaped because the peridium, when it breaks up, stays attached, and so you can just make out fragments of the peridium there. And then, of course, the spores are quite elegant. They have really large spines, ornamentation, and the spines arranged in these rows, which are almost forming a reticulated pattern. So it's a group that's under revision. There's several species that aren't officially published yet, but I'm still calling mine that, and we've got 4 of those in BC. And this is a nice thing about Meroderma. This is where you find it. This is up in northwestern BC. And the order Staminatidales, only 2 families, 8 genera, 23 species found in BC. The family Morocataceae, which I think is only recently erected in this with this grouping, but this is what we're calling it now. And we've got representatives of every one of these, I believe, in BC, and same with Staminatidaceae. McBridiola, named in honor of Thomas McBride, one of the founders of myxomycetology in British Columbia. And the very characteristic, very common one here that people can identify if they see it in this stage, Antherthenema papillatum has this disc at the top. And so the capillicium remains attached internally to this disc. They look like little eyeballs. Maybe we'll call that one the eyeball slime or something. And then another very common one, Comatrica nigra. Google has a really nasty translation of. Anyways, within the order Staminatidales, I chose Brepheldia maxima as an example because this one I just had some cool photos of, and it's also really common and often mistaken for many different things, for Fuligo or some other large showy slime molds. These things they can form massive plasmodia fruiting bodies that take over a whole stump. They like old stumps and big old cottonwood trees. Here's one in an old growth cottonwood stand up in the Slocan Valley, and it was just crawling up and around through the Zygus tessellatus is the mushroom, and you can see the remnants of the hypothallus in there. So I collected this and along with the Hypsozygus and brought it home and looked at it closely, and you can see it's made up of these almost individual sporocarps, and it's really the giveaway for oh, some weird sounds. Is the capillicium forms these lantern patterns. Checking in, are we all still good, Svims president?
Yeah, we're good. I just muted them. It's all good.
Okay.
Yeah.
No worries. These things happen. Okay, so there's Brepheldia. It's got a common name. I forgot what it is. We're working on that too. Apparently, the government wants us to put common names on all the slime molds in British Columbia. Andy probably has some experience with this, so it's kind of funny to come up with a common name for something that's been found once. Anyways, Brepheldia is quite common, so we could probably come up with a common name for that one. This Comatrica fusiformis, again, was a species that is a high elevation nivicola species that I got the first record for Canada well, the royal Ryan and myself were up in, again, doing some TEM mapping in near Revelstoke, and in some high elevation old growth habitat, and we found this, and it took me forever to figure it out, but there's only one Comatrica. Comatrica is known for having a evanescent iridium that just disappears. You never see it. It doesn't last. And here's one that had this persistent iridium, so I couldn't believe it was Comatrica. I went through many different things, and then asked Edvin Johannesson from Norway, who's one of the world experts, and he assured me that indeed that's what it was. And I've since found it a couple times, so quite confident that's what we've got here. And again, just who wouldn't want to go study these things? Where you get to go. These are great places. You don't have to find them everywhere, but anyway, most people go up to these places and they just stand around gawking at views and looking at wildflowers. But you poke around in their logs, you find that's where the real cool stuff is. The smaller you go, the cooler it gets. That's what I found. Again, in the order, Steminatidales, we have Steminatopsis typhina. Here's a very common species that goes through, as they all do, quite a few different changes from these little lollipops to this became quite a popular photo on Facebook because it's so easy to anthropomorphize. But to me, it's a murder scene. There's a dead guy there, and they're consoling themselves, and then of course there's this guy back here being interrogated. What did you see? Who did it? So I like that about slime molds. They have a sense of humor. They always gather together in groups, very party oriented. Now we get into the namesake of the Steminatidaceae, the Steminitis species. These guys, they make their stem first, and then they make their gloopy bits turn into spores on top. And so here's showing some of the patterns of development. And they start off as just little blobs of little spherules that then elongate. This can happen in hours, and then they go through these other changes as they mature, and eventually they turn into these the chocolate tube slime molds is what the common name for these guys is. There's a little beetle cruising away from the scene there. Find that there's some beetles that only live in the Steminitis, and they lay their eggs that develop inside this network cage inside them. Once the spores go out, and I guess in exchange, the spores get dispersed by the little critters. And just to wrap up with a few of my favorite critter slides, the globular springtails. You'll see a lot of those as you start to look closer at slime molds. Collembola. Here's one that was shy, and he disappeared into the slime chasm, but to come out again, and hard to catch a good photo. Is good all around as the Olympus TG series cameras are. They kind of lose it at the sub-millimeter level. I just like this photo as a kind of good example of the microcosmos that is there. We have our invasive little chocolate Arian slug, millipedes and centipedes and all sorts of mollusks and things, and it's just this whole living world, and a lot of these critters feed directly on the slime molds. Here's a little glass snail just cruising across a sea of trichia. And then some resources. I know it'll come up. Where do you get information on these guys? Well, first of all, citizen science. I totally took me a while to join the whole app world of let's shout out what we do. But the INAT is turning out to be a really cool resource. So we started this myxomycetes of British Columbia group, and I use it just to keep track of all my own observations. We can download our data. We can help others identify what they find. And so here we are, we're up to 142 species. May or may not be all right. There's a lot of missing identifications, which we try to sort out. But it's one way to share and learn about your own findings. And then pretty much my only reason for using Facebook is the slime mold identification and appreciation book group. Just a wonderful group of knowledgeable people, amateurs. Hasn't gotten all it's getting there, but it hasn't gotten as like goofy as the mushroom groups. No offense. I mean, Svims is wonderful, but in terms of like, they're just polite, and it's also worldwide. So that's one reason I'm a big fan. Oh yeah, okay. Well, I'll show you this. I just thought it was kind of neat. This picture of this tree, and I was thinking when I was looking at a Lamphoderma under the microscope, it's just like a tree. And that is end of my slideshow.
I got a question for you, Tyson.
Yes.
Is there for doing DNA work on the slime molds? Is there a reliable barcode region over all the slime molds?
Oh, yeah. it's the rDNA, IPS. I don't know. I haven't. I don't work in that world. So I will one day. I know, but there is, and it's different than fungi. So if you're familiar with the regions, I know it's the rDNA and or RNA, and it's in some papers, yes. And it is a distinct region. That's
Okay.
the best I can answer.
I was just wondering if there was a barcode.
Yes.
Any more questions at all? Anne, was there something online that people were asking, or were they just sharing a bunch of photos?
No questions from the chat that haven't been answered. It looks like it was mostly sharing of photos.
Yeah, that's good. Looks like
I
a
have
lot
a
of people.
question.
Yeah.
Well, I'm curious if others are collecting or photographing slime molds, because you're the people who see them, the mushroomers, and I guess it's more of a request. Is if you do, would you please upload them to the iNaturalist myxomycetes of BC group, because we really want to build our inventory, and it's
Yeah.
cool to find where people are finding stuff.
A good point. Definitely finding some, but not uploading them to iNaturalist or anything. So I'll get on it.
Looks like we're getting a couple questions in the chat. We have any good resources out there until the new book comes out?
Yeah. Sorry, I've lost. Oh, there's stop share. Okay. There, I couldn't. All the buttons change after I lost my internet. You know
That's
how
all
that goes.
good.
So there's the chat. Yeah. Okay. Any more good resources? Well, no. I mean, there's very few regional guidebooks. There's, at least for identifying, I use this is my Bible, which is going to show it backwards on there, but Les Myxomycetes, which is the French book, which is out of print, which I was able to find. That's from 2011. There's the German book, Des Myxomyceten, which is probably still in print. It's good and comprehensive. And then there's the Dutch book, Nenenga Brennenkamp were the authors. And again, they're good in that they have keys. Another good one is Bruce Ing's book, The Myxomycetes of Great Britain and Ireland. And that one you could probably still find copies of. It was more available and cheaper. I got a copy on Amazon. And Sarah Lloyd's book, Where the Slime Molds Creep, and she's from New Zealand, well, from Tasmania, actually. Not so much a comprehensive ID book, but good informative book. and then I would recommend online sources. It's called, there's the University of Arkansas has a page. I can put the link up in the chat. Actually, that's what I'll do. I think that'd be the easiest way, which is nomen myceta. Mycetozoa, that's what it's called. And then beyond that, we're building a extensive library of all the individual papers and things like that. But it's not like there's one, mushrooms of British Columbia book with some nice photographs that I can just flip through and go, oh, and then here's how they're all organized and some information, maybe a little key. That's a good idea. Maybe we should write a book. So that's kind of where we're at. There really aren't very many regional guidebooks, like those three that I mentioned. Four, I guess, the UK, the Dutch, and the German and the French ones, and they're all out of print.
Natasha, I noticed that you have a question. Do you want to ask that?
Yeah, that was a great presentation. Amazing photos. So thank you. I missed, sorry, my dog. I missed like the scale of a lot of those photos with, like, the really beautiful globular round shapes. Like, sorry, my terminology. But what's the scale of those photos, and like what kind of a lens are you using?
Sure. No, other than the pictures, a couple of them in my hands, I'm guilty of not really taking good things that show scale, but we're dealing in mostly in less than 5 millimeters. So what's the smallest mycena you've ever found? Kind of in that range, where you want kind of 2 to 4 times magnification to really get an appreciation of them. And that's what the cameras that we have in the field, the Olympus TG series, these little, I started with the TG3, I think, and I'm up to the TG6 now. They have a light ring on them, so and their image stabilization and macro photo stacking built-in capabilities are ideal for my uses. I used to spend a lot more time and money on equipment for photographing, but then I would spend more time photographing and less time looking for stuff. And there are people that are just so good these days with macro photography that we'll send them our specimens and get them photographed. So, but yeah, we're dealing, Natasha, in really tiny things. Like, I find some species only by scanning through the stereo microscope. But many of them aren't that small. You can see large fruitings when they're in their plasmodium, even over a meter long, taking over a log. Those are the eye-catching things. But most species are tiny, less than a couple millimeters. Yeah, I will find the sharing thing.
That was brilliant, Tyson. Thank you so much.